Everyone, my name is Joshua in Quinit. Today I will present to you my review paper entitled A Review of Biomimetics in Tissue Engineering, Facile Physisorption of Polydopamine. To begin, we start by introducing the concept of biomimetics. Otto Smith coined the term biomimetics in the 1950s to describe the study of biological systems, structures, and processes, and how products can be synthetically made by mimicking them. However, this is not the first time this approach has been used. People have taken a lot of inspiration from nature a long time ago. The field of biomimetics got its prominence because of the notion that there's no better model to create new things than nature. As you can see from the figure presented, biomimetics has been incorporated into a wide array of applications, including art, medicine, architecture, and engineering. As you may already know, there is no single definite scaffold that can be used universally. But unlike traditional constructs, biomimetic scaffolds made from natural biomaterials can provide cells with a wide range of biophysical and biochemical stimuli that resemble that of the native extracellular matrix, which is advantageous in regenerative medicine. Further illustrating this, let's take tendon tissue engineering as an example. In treating tendon injuries, biomimetic materials could render an extracellular matrix that replicates that of the microenvironment of the native tendon tissue. This enhances the efficacy of regeneration. It is pretty obvious that biomimetics transcends a multifaceted approach that could make outstanding platforms for tissue engineering designs. In the succeeding slides, we will focus and take a look at one of the most fascinating biomimetic materials that is currently being explored for its remarkable properties, and that is polydopamine. Polydopamine is a product of oxidation reaction that has been making waves in various industries, thanks to its impressive ability to coat different surfaces, not needing complicated chemical processes due to its self-assembling nature. PDA's muscle-like behavior enables it to stick to a wide range of configurable materials, such as ceramics, metals, and even polymers. This makes polydopamine a top choice for tweaking the surface properties of a hydrophobic polymer, which is vital for improving how cells interact with polymeric materials. There's actually a classical way of creating polydopamine films on surfaces. A substrate is first placed in a dilute, slightly basic dopamine solution exposed to the atmosphere. The oxygen then oxidizes the dopamine into dopamine quinone, which begins a complex reaction, eventually forming a quinone intermediate, and triggering the polymerization and self-assembly process, which then produces the PDA. Adjustments in the deposition time or the concentration of dopamine in coating solution, which needs to be above 2000 parts per million, can help achieve the desired polydopamine film thickness on surfaces. Based on the previous slide, we know that PDA undergoes several initial reaction phases. However, the culminating steps leading to PDA formation are still controversial. These meet several hypotheses about its nature. Still, polydopamine is believed to be polymeric, as depicted by the carbon coupling along its dopamine monomers. Succeeding research works relating to functional groupings and how these groups fluctuate during the polymerization process are imperative to clarify the exact mechanism of polydopamine assembly. We have here three of the most investigated characteristics of polydopamine. In terms of hydrochemistry, polydopamine is able to diminish the water contact angle of a myriad of surfaces and enhance the wettability of biomaterials to improve cell uptake and cellular transduction. Another characteristic of polydopamine is its ability to change its charge by applying pH stimulus. The reversible selectivity of polydopamine to cations and anions is due to its pH-sensitive charge. Last on this diagram is polydopamine's reactivity. Due to the presence of its catechol groups, polydopamine is able to provide nucleophilic interactions of quinone with amine and thiol molecules. This is also the reason why polydopamine can control the immobilization of peptides, growth factors, or other biomolecules, and also attach itself to both organic and inorganic surfaces. In the realm of tissue engineering, functionalizing surfaces usually relies on chemisorption, which involves forming a chemical bond with the surface through covalent interactions. Despite its specificity and robust bonding mechanism, it often needs arduous surface treatments that may induce toxicity or instability. On the contrary, physisorption uses non-covalent interactions like hydrogen bonding and etc., which are simplified, more flexible ways to modify surfaces. This is especially advantageous when applying PDA to biointerfaces, as it enables the creation of coatings that can be easily applied, removed, or altered under mild conditions, thus minimizing the need for harsh chemical processes. Of course, there are still challenges that need to be curbed. PDA coatings, especially those applied through physisorption, may be less stable than chemisorbed ones. Physicochemical factors such as pH, temperature, and ionic strength can affect the reliability of PDA coatings over time. Moving on to the applications of polydopamine in tissue engineering, polydopamine has been used to enhance cell adhesion. When the contact angle is low, commensurate to high affinity to water, it generally needs to improve cell adhesion and protein absorption. The efficiency of surface sensitivity determines the scope of resulting cellular activity, and so we have here the Berry limit, which is an archetypal term used to describe the threshold for biomatter adhesion, which is equivalent to 65 degrees. It also refers to the point at which surface can be viewed as optimally hydrophilic. So on this account, various tactics are used to fine-tune cell adhesion on different surfaces. As most polymers used in biomedical applications are either hydrophobic or superhydrophobic in nature, there is a need for coatings that reinforce wettability to further facilitate hydrophilic patterning on polymeric surfaces. By reason of biocompatibility and fluidity to adhere on any surface, polydopamine became one of the interesting biomaterials for surface tuning for both micro and nano drug carriers. Apart from inducing cell uptake and mass transport, PDA can regulate the release of drugs at specific sites in vivo, in most cases in response to external stimuli. When electrical stimuli, for example, were combined with PDA's exceptional properties to coordinate cell affinity, the resulting structure yielded a dynamic environment that is conducive to cellular growth. From optimizing its design and synthesis to uncovering latent applications for musculoskeletal system regeneration, polydopamine piques scholars spanning disciplines in the effort to stir up functional recovery from bone and muscle disorders. As with other research, the use of polydopamine in tissue engineering poses concerns and limitations. The previous chapter unveiled multifarious opportunities for polydopamine in the paradigm of tissue engineering, but still, um, the stability of polydopamine is yet to be uncovered. In line with the progress observed in polydopamine research, the next phases of this muscle-inspired approach to tissue engineering can be reckoned on the refinement of synthesis and post-processing techniques, which will attest to the stability, reproducibility, and scalability of applications. Irrefutably, novel methods to modulate the rate of PDA degradation in vitro and in vivo are also crucial. So in a nutshell, we were able to cover a significant portion of biomimetics in tissue engineering, proving its versatility in tissue engineering specialties. Self-assembling PDA nanoparticles unlock prolific opportunities to advance medicine and healthcare. There are still untapped areas in this space that future research can exploit, but most of those are explicable limitations in stability and scalability that can be resolved by the design of high-throughput visible synthesis of this biomimetic material. Thank you so much for listening.